experimental protocol, which was to construct our suicide vector consisting of our ECR, toxic imaging, and an ampicillin resistance gene. So now we have to move on to step 2 of our experimental protocol, which is to construct a destination vector, which consists of ECR, minimal promoter, and our GFP gene.
Destination, because it's destined to go where?
It's destined to go to cells in the chick spinal cord neuron.
All right.
He's going to get to that.
So 10 days, sadly, was not enough to put both of our ECRs into destination vectors. So what we had to do instead was use the ECR MM123 in our destination vectors instead of the ECRs that we prepared personally. MM119 and MM148 were the ECRs that we prepared personally. In the future, they'll be put into destination vectors by future CMB interns to be used by future CMB students. In our case, however, we're using MM123. It was prepared by former Neurolab students, but interns were able to put it into a destination vector and have it ready for us in time for our microinjections. So the interns at CMB started off with our suicide vector. They used restriction enzymes to cut it out of the suicide vector, leaving us with just the ECR with the adapters on the ends. Because the ECR has the adapters on the ends, it easily attaches to our destination vector, which has spaces for the adapters. So now we have our destination vector with ECR, our minimal promoter, and the GFP gene. But we don't have enough of the ECR to do a successful microinjection into chick embryos. So what we had to do is use the technique of bacterial amplification laid down by my colleague Sarah. So the interns put in the destination vector into a bacteria, and the bacteria was allowed to proliferate and replicate on a special growth medium. Once the bacteria replicated, the destination vector replicated along with it. So eventually we have a sufficient quantity of destination vectors to be used in our microinjections. So we've just finished step 2, which is the construction of our destination vector. And now we're going to move on to step 3, which is to microinject our destination vector and indicator dye into the neural tube of chick embryos. And then we're going to go on to use the technique of electroporation to get the spinal cord cells to take up this destination vector.
Vic, you're probably going to mention this, but for the benefit of the audience who may be unfamiliar with the term neural tube, can you just tell us what that is?
So the neural tube is a hollow area right here, as depicted. Eventually it's going to form into the spinal cord of the chick.
That's all.
So we inject our destination vector and indicator dye into the neural tube of the chick embryo. We use indicator dye because we need to see that we injected our destination vector into the correct region of the chick embryo. If we only injected our destination vector, we would not be able to see where we put it because DNA in a liquid naturally is transparent. So we would have no idea whether we got our DNA into the right spot. We inject our ECR into the neural tube because the ECR controls gene expression in the neural tube, as shown by scientists at the Lawrence Berkeley National Lab. So here's a video of my colleague Sarah performing this microinjection on a chick embryo. So as you can see, this right here is going to become the head. This is the beating heart. This is going to become the tail. We call this the caudal region. And so Sarah's preparing to inject it right now. So she punctures the neural tube with the micro injection needle. And as you can see, the indicator dye and the destination vector travel from the caudal region all the way up to the hindbrain of the chick embryo.
I got it.
Show it again.
Go back for a
Show it
second.
again.
Show it one more time.
Was it just Sarah doing this?
No, we all did this
You all did
actually. We were all successfully able to do this.
This is
all
just
10
a
of us.
representation.
Yes.
Sarah was quite good at this.
I have to take a moment to brag for the benefit of the audience, because this in vivo assay, to gain proficiency with this in vivo assay can take many graduate students a week or more of time to be able to do an injection. This is a perfect injection. I have done thousands myself. Ralph has done many more than that. Could not do a better injection than this. And this is with 2 days of practice. 2 days of practice, on the third day the experimental injections, and all of them gained near perfect proficiency with this technique.
If we had a week, they'd all be experts, and I'd have you guys come into the lab, and I'd drink coffee and move on to more lofty goals while you did that. But yeah, I mean, really, fantastic.
That is nothing short of exceptional, and we're really proud of you guys and impressed.
All right, so moving on, we now have our destination vector inside the fluid-filled space of the neural tube, also called the lumen. But we need to get our destination vector inside the spinal cord cells of the chick. So we're going to use a technique called electroporation. Normally, molecules with an electrical charge can't pass through the phospholipid bilayer of cells. But electroporation uses small electrical pulses to form these transient pores across the phospholipid bilayer. This allows for our destination vector to easily come into our these spinal cord cells. Now I said that these pores are transient. That means that they're temporary. So eventually the cell will close shut and we'll have our destination vector inside of our cells.
What the hell are those 2 X's there?
That is the chromosomal
Okay.
DNA of the cells. Okay, so here is a top-down view of the electroporation. So we have a positive electrode and a negative electrode, and they're placed near the caudal or tail region of the embryo. This is because we only want to visualize gene expression in the caudal region. So to get these cells inside of the spinal cord cells, we can take advantage of DNA's slightly negative charge. Because we're using a positive electrode, once we introduce these small electrical pulses, the DNA is going to be attracted towards the cells nearest the positive electrode. So it is going to move from the lumen into these cells on nearest the positive electrode. This is a cross-section of the neural tube following electroporation. As you can see, indicator dye is in the lumen, and we have our destination vector on the cells nearest the positive electrode.
Show us with your hand what a cross-section would be.
So a cross-section, we would cut right here and then invert it.
Cut this way and looking this way.
Yep.
I have a question. Go back one slide for a second. So Ralph asked what those are. Those X's are the chromosomes. We've used a neat little trick to coax our developing spinal cord cells to take up an introduced plasmid. Is that plasmid getting incorporated into the chromosome?
Yes, it will be.
Really?
No, it's
You
getting.
guys wanna help?
We may not have covered it. It's not gonna get integrated into
Well,
the.
wait. So when we talked about genetic versus non-genetic model organisms, first question, I know you know the answer to this. Is our system, is the chick model system a genetic model organism?
No, it's a non-genetic model organism.
Why is it a non-genetic model organism?
Because any genetic modifications we make to the chick won't get passed down to the offspring.
So I ask you again, is our plasmid getting integrated into the chromosome?
No.
No, it is not, and it doesn't need to be, because we're just looking at transient expression, or the possibility for transient expression. This is a neat little trick that is a function of the system, and is part of the reason why we chose the chick model system for these experiments, because they can be done quick, dirty, fast, and you can look at transient expression. There are other techniques, not in the chick, in other model organisms, where an introduced foreign DNA will get integrated into the host organism's chromosomal DNA. We don't have to do that here.
We don't want to
We don't.
maintain colonies of chickens, though, do we?
No, we
based
don't.
on some of the conversations we had. So Vic, you may have mentioned this already. Go ahead, you can go to your next slide. So in how many, did you say this already? In how many cells, theoretically, should the plasmid be introduced?
So the plasmid should be introduced into all of the cells nearest the positive electrode.
Okay, and what would be, again, and this was covered before by one of your colleagues, what would flip the switch on that plasmid and drive GFP expression?
Transcription factors.
Okay, in every cell?
Not in every cell, and only the cells in which the transcription factors are present.
Great, okay. I'm sure Leanna may want to go there again.